Hello everyone, welcome to another biology class by Online Biology Tutorials. In this video we will study transcription, and transcription is the process wherein messenger RNA is synthesized from DNA. Okay, DNA we already know it is deoxyribonucleic acid. RNA we already know it is ribonucleic acid, and here this small m refers to the messenger RNA, which is a type of RNA. In this video itself we will be talking about different types of RNA as well. So stay tuned till the end, because all the terms which I am going to explain, all the terms are important. They may come in the short question, it may come as a long question, they may ask you for the enzymes. So everything will be included in this video. So DNA, you know, it is double-stranded. One strand is 5 to 3 prime and the another strand is 3 to 5 prime. In my initial videos on DNA, I have already explained why the two strands are anti-parallel, so do watch it out. One strand of DNA, which is 5 to 3 prime, it will be known as coding strand. And what I told in my previous video, that coding strand is also known as sense strand, whereas the other strand, which is 3 to 5 prime, it is known as template strand, and it can also be known as anti-sense strand. Now here it is very important to know that coding strand is a one whose sequence is exactly same like that of the messenger RNA. The only difference is in the T, because there is no thymine found in the case of messenger RNA. So in case if it is thymine is there in the coding strand of DNA, it is going to be U or uracil in the messenger RNA, right? So coding strand and messenger RNA, they are going to have the same sequence. Because they are going to have the same sequence, that is why coding strand is also known as the sense strand, because the sequence of bases, the senses are same in both, only with the difference of T. T of DNA is replaced by U in the case of messenger RNA. Now if I say about template strand, this is the one from which transcription will start. Okay, this is the sequence which is going to get transcripted, right? So messenger RNA and template strand of DNA, they are complementary to each other. Means if it is A in the template strand, then it is going to be U in the messenger RNA. If it is going to be T in DNA, it is going to be A in the case of messenger RNA. If it is C in the case of DNA, then it is going to be G in messenger RNA, right? So the bases are complementary to each other. That is why template strand is known as the anti-sense strand. Means the strand of this sense is 100% complementary to the other strand. Okay, so what we have learned more, that the messenger RNA will have the same sense as that of the coding strand. There would be a difference of T and U. And another thing we have read, that template strand and messenger RNA, they will be complementary to each other. Now because messenger RNA is synthesized from the template strand, because they are complementary to each other, so in case here it is 3 prime N, so here it would be 5 prime N. Here it is 5 prime, so here it is going to be 3 prime. Remember, synthesis always has to happen in 5 to 3 prime direction for messenger RNA, and in the case of DNA replication, it was already stated how it happens. So do watch out a video on DNA replication as well, because then the things will be much clearer to you. These are the two strands of DNA which I have drawn. Of course, one would be 5 to 3 prime and another would be 3 to 5 prime. Now the bases in messenger RNA, they will be complementary to the template strand. but both strands of DNA, they are going to help in the transcription process. Okay. Now, before I start, let's know what is transcription unit. Okay. Transcription unit is actually a part of DNA that is going to take part in the transcription process. Okay. The segment of DNA that will be transcribed, it is known as transcription unit. And what are the components of it? So question may come in the board examination. What are the components of the transcription unit? So they are 3 in number. Just watch very carefully on my screen. What am I going to do? So this is the P, this is T, and this is a flanking region in between, which is known as structural gene. Okay, this is our structural gene. So here we will write P means promoter. Okay, P means promoter, T means the terminating sequence or the region that will help in the termination, means cessation, means it will bring an end to the transcription process. Okay, so if certain part of the DNA has to undergo transcription, then there is a must requirement that that transcription unit should have a promoter, a structural gene, and a terminating gene that will bring up the termination process. Okay. Another thing which is very, very important here is transcription will start from this point. So this point, which is the starting point of the structural gene, this is the point where the transcription will start. But prior to it, you can see that promoter is also there, right? So the sequences that are occurring prior to the start point, they are known as upstream sequences. Okay, and the sequences that are near the terminating sequence, they will be known as downstream sequences. Okay, downstream region also we can also say like that. Okay, in certain books they have mentioned, this is our coding strand, right? In certain books they have mentioned that upstream region is a one which is close to 5' end, and downstream region is a one which is close to 3' end. So that way also it is correct. There is no mistake in it. Okay, so transcription unit is a part of DNA that will undergo transcription. If it is undergoing transcription, it should have 3 parts. One, the promoter, another, the structural gene, another, the terminating sequence or the termination site. Now let's discuss about the enzyme that is going to participate in the transcription process. Okay, so the enzyme that is going to participate for the transcription process, it is known as RNA polymerase. This RNA polymerase, it is not synthesizing on its own, rather it is dependent upon DNA. So we can call it as DNA dependent RNA polymerase, right? This way we name it. Now we have 2 types of organisms, as you know, prokaryotes and eukaryotes, right? And we have different types of ribonucleic acid in our body. This could be ribosomal RNA. So remember, whenever you are going to write R as short in caps off in small letter, because this R is denoting ribosomal. Okay. The second type of RNA is messenger RNA, and here M stands for messenger. M here also has to be written in small. The another type of RNA is tRNA, and T here stands for transfer RNA. Okay, there is another type of RNA which we have to study, and this is snRNA. This is also important to know, and we will be telling its importance later. This is a small nuclear RNA. So this is our RNA, forms ribosomal RNA, messenger RNA, tRNA and snRNA. This much we have to definitely study for our board examination. Okay. Now we have said that organisms are of two types, prokaryotes and eukaryotes, right? So in prokaryotes, there is only one type of RNA polymerase or DNA dependent RNA polymerase that is going to synthesize all these different types of RNA. But in eukaryotes, there is a difference. We have different types of RNA polymerases that help out in this process. So this is important to know that ribosomal RNA is synthesized by RNA polymerase 1. Okay, remember, whenever you are writing 1, it has to be written in the Roman numerals. Here it would be RNA polymerase 2. Okay, and the third type of RNA polymerase is RNA polymerase 3, which is going to synthesize transfer RNA as well as small nuclear RNA. Now let's study the structure of RNA polymerase, which are the components present in it. Okay, so the very important part of it is the sigma factor. It is represented by the sigma, and the second important part is the core enzyme. The core enzyme, of course, when the name enzyme is there, that means it would be proteinous in nature. And you can see that 4 polypeptides are being shown. 2 are similar to each other, and that's why I have written alpha for them. So we could have written like this also, because beta and beta dash, they are this similar. So we can write alpha to beta b dash also. Okay, so here this role of sigma factor is very important. What does it do? Sigma factor identifies a specific sequence in the promoter, right? So there is a specific sequence in the promoter, and this makes sigma factor to bind to the promoter sequence. As soon as sigma factor binds, these sequences, the core enzyme is also accompanied. And once these are there in unison, means when both the parts of the DNA dependent RNA polymerase are there, then functional enzyme is formed that could do the process of transcription. So these parts are very important. Sigma factor is identifying the promoter sequence, and then comes the role of core enzyme. Okay. Now, as I said that promoter has a sequence, a specific sequence, we will be talking about it. We have already seen that promoter sequence is a one which is located upstream, just prior to the start point of the transcription. Okay, so promoter region has specific sequences that will initiate the RNA polymerase to do its part, because this is the one enzyme that is to do action. Okay, so promoter sequences have a special sequence which is known as Tata box. Here TATA, you can see that it is written in the capital letters. Here T means thymine, A means adenine, thymine and adenine. Means this will be a specific sequence that will have T and A rich in there. And this sequence was discovered by Pribnow scientist. Thus, to honor him, we can also call it as the Pribnow's box. Generally, the sequence in the Pribnow's box is Tata AT, right? It is known as Tata box. It can also be known as Pribnow's box, but the exact sequence is Tata 80. Right? Now you can see that all the sequences, they are containing either T or A. Why? Why only T and A are present, not G and C? The reason behind it is that T and A, they contain only 2 hydrogen bonds. So it would be quite easy for them to get break up. But G and C, they are held together by triple hydrogen bonds. So they would be requiring more energy for the breakage. So with this, it would be quite easy. Next, we will be discussing in the later video. Stay tuned for part number 2 of transcription. And if you haven't subscribed to my channel, please do it immediately. Thank you.